Welcome back. And so our next speaker will be Raphaël Rodriguez from Institut Curie with chemical control of cell adaptation. The floor is yours.
Thank you, Hugues. Thank you also very much. I am very happy to be here with my friends. I seem to be the new kid on the block in this area of redox biology and cancer. All right, so before I start, in the interest of time, I want to acknowledge the talented people I've been working over the years in my lab. I've been fortunate enough to meet these people and many collaborators, too many to be listed, too many that are instrumental to the work that we have been doing over the years. All right, so what my lab is working on is this idea of cell adaptation, the fact that our cells have the capacity to adapt, to change between cell state, phenotype, adopt a new identity. It is also called cell plasticity or cell state transition, and we have chosen to use the wording cell adaptation because it better reflects the idea that a cell is going to adapt as a result of something. It can be a physical stress, mechanical forces, for example, during development. It can be the presence of a cytokine or growth factor in the tumor microenvironment, for that matter. It can be the presence of a pathogen that will force our immune system to respond. And for me, and in cancer therapy, it can be the exposure to chemotherapeutic agent. And we know now that drug tolerance is acquired mostly as a result of non-genetic mechanisms that are dynamic and reversible. And that's how we know they are not genetically driven. And so the idea is that if you understand the chemistry underlying this cell adaptation, you can identify new vulnerabilities. And if you have that in hand, then you can design new drugs and new treatment. And so our modest contribution to that space is to have shown that metal ions regulate cell adaptation. In fact, there is no possible adaptation without metal ions, two in particular, copper and iron. And in biochemistry, in textbook, you will find that metal ions are very often coined as cofactors, and it is mostly true, except for copper and iron, which are different. Why are they different? Because they can capture or release an electron. And so if you see electron motion, it means a chemical reaction is happening. And in that case, these are no longer cofactors, they become master regulators, they become catalysts of redox chemistry. And as a result, they do have a rate limiting role in the cell. So it's not whether or not cells have these metal ions, it is the extent to which you will see a shift in the amount of this metal that you have for a specific process. And we go back to Le Chatelier. If you have an equilibrium and if you want to shift your equilibrium toward a state, you need to feed more reagent. So you need to feed more catalyst here. And so my claim today is that metal ions, copper and iron, are just as important as transcription factors, epigenetic writers, readers, and erasers. So now, studying metal ion is difficult because you cannot knock in a metal. You cannot knock out a metal. Metal ions are tightly regulated. You have iron import proteins, you have iron export protein, you've got iron storage protein, you've got iron trafficking proteins, and then you've got all the protein using these metal ions as cofactors or as catalysts. But not only that, but two other things. Metal ions are a lot smaller, so the diffusion rate is a lot faster, making them more difficult to study in time and space. But also they do exist in specific forms. They can be bound, they can be free, they can be as a +2 redox state, +1, +3. And so if you want to study metal ions, you need to design probes and compounds for each of those specific degree of oxidation in organelle-specific manners, which makes it a lot harder. So this is a landscape, as we know, iron homeostasis being regulated. And you need to take a mental snapshot of this. And now this is how copper is regulated. And if you compare the two, they are mostly completely different. And yet, when you look at the periodic table, you will see copper being not too far from iron. So you see that a few protons, mostly 2 protons, completely change how a cell deals with these metal ions. Now, I am not an inorganic chemist. As a matter of fact, I did train in Marseille, and my PhD advisors then used to say, even an inorganic chemist would understand, just to convey the idea that inorganic chemistry is not worthy of your time. I respectfully disagree. It is just to illustrate that this is not my background. So the question is, how did we end up working on iron and copper biology? So when I did start our lab, it was 2012 in Suivez-nous, Natural Product Institute. I was eager to do something that I had never done before, to be very distinct from my prior mentors' expertise and what I had learned from them. And so I did stumble on that paper from Bob Weinberg, Piyush Gupta, and Eric Lander that they did a screen and they identified a natural product which in the cell is capable of eradicating what we call tumor-initiating cells. They called it cancer stem cells, but what it means is you have a specific population of cells capable of initiating a tumor, and these cells are known to promote metastasis and are also known to be naturally drug tolerant. Now that paper was published at Cell, it is cited 4,000 times, and it was instrumental to me because for years as a chemist I was taught that we need to develop drugs that are more potent than doxorubicin, more potent than irinotecan, more potent than etoposide, more potent than cisplatin. All of them target proliferation. And then when you read this paper, you understand that it's not a matter of making them more potent because the cells that are responsible for metastasis and drug tolerance do not divide as fast as the others. And so you can improve Taxol as much as you want. You can make a super effective Taxol. This is not going to solve the problem of acquisition of drug tolerance. And that paper shows that cells can have another identity that require another class of drug. That's why it was instrumental to me. Now the paper is very interesting. They identified this natural product, salinomycin, and they wrote this text, and there was one sentence in that big paper that caught my attention. I thought, this is weird. Further studies will be required to characterize the connection between potassium membrane potential and cancer stem cell biology. Why is it weird? Because salinomycin was co-crystallized with potassium. These are ionophores. They form a lipophilic shell around the metal, and these complexes are diffusing passively through membrane, and as a result, if you use a lot of them, they will increase the concentration of potassium in the cell. This leads to depolarization of the outer mitochondrial membrane and induce death. But this is true for all cell types, whether these are cancer stem cells or normal proliferating cells. And so the reason why these compounds were killing selectively cancer stem cells did not add up to me. Why would the membrane potential be differently regulated in the stem population compared to the others? It would be like me telling you the rules of gravity are different in the moon and on Earth. That's how I read it. And so we decided to study. So why did he say this? Because he thought that the mechanism was involving potassium because of the crystal structure. So note to yourself, a crystal structure almost never explain a mechanism of action. You need to keep this in mind. We were fooled so many times. So at that time, and I pointed out this was published 6 months before the work of Vasanti and Stewart on the role of JPX4 in persisters. We discovered that these, making more potent analogs, these natural products accumulate quantitatively in the lysosomal compartment. And knowing a little bit about iron biology, we knew that iron is trafficking through the lysosomal compartment, and we proposed that perhaps the mechanistic target was not sodium, potassium, but iron. And so just by looking at the iron content in the lysosomal compartment of these cells, we found that it was sequestered. And in fact, if you have a lipophilic counter-ion that is tightly bound to it, what this compound is doing is reduce the capacity of the metal to escape the lysosomal compartment. And over time, you will see an increase of the concentration of iron in the lysosomal compartment because it cannot exit. You will see occurrence of lipid peroxidation over the course of 48 hours, and your cells are going to die. At that time, we compared the stem versus non-stem population, CD44 high, which characterize stemness, and CD44 low, which characterize proliferating cells, and we found by mass spec that there was more iron in these cells. And so why are the stem population dying of this compound? Because there is more iron in them. And so if you want to kill your cells, you can promote iron chemistry, or you can suppress ferroptosis suppressors which scavenge those. Now, later on, we used sublethal doses of these compounds, and what we found is that JPX4, Marcus's protein, and XCT were upregulated. And so the cells can survive treatment by upregulating this protein. However, at that time, first of all, we did not want to use this word pheroptosis in the title. It was 2017, just after it was coined, because we felt that this had been defined by many people before 2012, specifically Marcus. But also that paper in 2012 discussed pheroptosis in the context of a KRAS mutation dependency, and all our systems were KRAS wild type. And so we wrote in the text a phenotype reminiscent of pheroptosis, which is iron-induced lipid peroxidation-mediated death. Now, that study raises two questions. Why do cancer stem cells or stemness require more iron? Question number one. And how do cells that are isogenic, the genome of these two cell lines is the same? How do they manage to upregulate iron uptake? Because if you look at textbook, you find that transferrin and transferrin receptors, this canonical iron uptake pathway, negatively regulate itself at the translational level because they are iron responsive element, which allows cells at the basal level to maintain an amount of iron that is low enough such that the cells are not dying of redox stress. But if the cell need more iron, how do they do? If transiently you need to catalyze chemical reaction, how do you override this mechanism? These were the two questions. Right, pass on that. Yeah, later on, many people started to work on lysosome, iron, and pheroptosis, including Michael at not the Rockefeller, but MSK. And he found that it is difficult, in fact, to identify. It was difficult to nail the role of lysosome in pheroptosis because simply when you lyse your lysosome, many things happen. So you have distinct phenotype based on the cell state, EMT state, and the cell cycle. And that's why it's difficult to nail a single homogeneous phenotype when you target this pathway. Now, this is what we have discovered, and this cartoon summarizes 15 years of my life past. We stumbled on this CD44 because it is a cancer stem cell marker, but also marks acquisition of drug tolerance. It was known for 50 years. And to me, the only very solid evidence of mechanism or involvement in biology is that it is a main receptor of hyaluronic acid. Hyaluronate are about 800 kilodalton in humans. They contain about 3,000 negative charges. And of course, because CD44 is recycling and the plasma membrane is not polarized, you would ask, okay, so hyaluronate, 3,000 negative charge per molecule is taken up. So what are the counter ions that keeps it neutral, such that the plasma membrane is just not polarized? And so just by inducing epithelial mesenchymal transition to have the two states, CD44 high-low, we discover that the main binders are calcium, manganese, but mostly iron and copper. And so when you induce your EMT, you will see upregulation of CD44, and you will promote this two-metal uptake. So we discovered two chemical reactions that are rate limiting. First of all, a new reaction catalyzed by copper-2, which in mitochondria, which was not known. Copper-2 activates hydrogen peroxide to maintain the NAD+ pool high. This is a co-substrate of many of the enzymes that are responsible for the production of ketoglutarate and acetyl-CoA. Those two metabolites traffic to the nucleus, and that will be used to acetylate histones and to demethylate histones. And so it is copper-2 in mitochondria that remotely controls the epigenetic landscape, the transcriptional profile, and the cell identity. So if you take out copper-2 in this process, you control cell adaptation. That was the idea. And the second one is that the rise of iron that we see in the stem state, iron is not mitochondrial as we expected. It is nuclear. We know this because we extract nuclei and we quantify. And in fact, by RNA-seq, we found that many demethylases that are iron-dependent were upregulated. So here you go. If iron is a catalyst, if you upregulate an enzyme that uses this catalyst, but the catalyst is not there, the enzyme is dead. And so you need to have upregulation of nuclear iron for these demethylase to be effective. 85 of those. Now, Bob Weinberg came 5 years ago. I took a screenshot of that slide, and Bob says the most reliable marker of tumor initiation and drug tolerance is CD44 across the board of solid tumors. And so that was we published our paper just at that time. Right, so a few data, and we go quickly on those. So we use this HMLER cell line, which reflects stemness. So P53 is out, H30 is up, E-cadherin is taken out, and you have the Kirsten-Ras mutation. You take out CD44, you take out transferrin receptor. We use RONOX-M, which stands for chemically reactive iron 2 in the lysosome only. And we found that there are actually two mutually exclusive iron uptake mechanisms that nobody knew about. It was all about transferrin receptor, but we find that there is a hyaluronate dependent pathway. If you take out CD44, that population drops. You have remaining signal because you overload your cell with hyaluronate. There is a remaining fluid phase on the cytosis. Same is happening with transferrin receptor. Now, these are chemically produced reagents. What about endogenous protein that are important for iron homeostasis, ferritin in particular, the iron storage protein? You feed your cells with hyaluronate. In the wild type, you have an increase of ferritin, meaning more iron. That increase is not detected when you have CD44 that's out. And now when you do this in the transferrin receptor knockout, you see this increase as well. That's the how. What about the why? This is a mass spec experiment that compares upregulated versus downregulated protein in the mesenchymal versus epithelial cell state. So those are the proteins that are upregulated. And we were happy to find PHF8. So this is a triple negative breast cancer cell line, but we found this in lung, pancreas, and so forth and so on. PHF8 is an enzyme that uses iron as a catalyst, and this is a catalytic cycle, and alpha-ketoglutarate as an electron donor to demethylate histones. Its main target histone mark is H3K9 dimethyl, which is a repressive mark, meaning that if you take out this mark, you unlock the expression of your gene. Now you induce your EMT with EGF, you see CD44 going up, textbook, and you see H3K9 dimethyl going down. Why is it going down? Because at the same time, you feed your enzyme with metal catalyst so you can take it out. Now if you take out CD44 at the plasma membrane, you block this demethylation in the nucleus. And so we like this data because it links the protein to the nucleus through iron trafficking. Now this is a catalytic cycle that illustrates the fact that iron is a metal catalyst, and here the co-substrate or the cofactor of this process is the enzyme. So if I take a flask, use iron only, oxygen, and alpha-ketoglutarate, I will see demethylation in the flask. And so you might ask, why on earth would cells evolve 85 different iron-dependent demethylases if you can do the job without? For two reasons, because the catalytic turnover is a lot faster with the enzyme because you stabilize the catalytic core. But most importantly, the enzyme is what defines the gene loci that is going to be targeted. Not all the genes are demethylated when you upregulate CD44. Specific genes are going to be targeted, and some others are not. Now, my claim is that iron is a master regulator of cell adaptation, and you should rightfully so challenge us on that. And to illustrate that point, we did what we call an EMT-MET transition, whereby you can treat with EGF, you induce your shift, and then you remove EGF and you go back to the epithelial cell state. And you have three pools of enzymes, all iron and alpha-ketoglutarate dependent. Some require to maintain the epithelial cell state, a second pool to promote EMT, and a third pool, different from the first one, that promote MET. And so the common denominator here is iron. If you remove iron, there is no plasticity at all. So to adapt, any cells need iron. Now we chipped H3K9 dimethyl to identify the gene target. You correlate this with the transcripts that go up when you lose your mark. And all these genes that were identified are associated to wound healing, EMT, development, cancer metastasis, mark drug tolerance, and our poor prognosis. All biological processes CD44 had been associated to. So if you ask me what is the mechanism of action of CD44, it is to promote metal uptake. Now, if you induce your EMT but you co-treat with deferoxamine, you hijack the catalytic center, which is iron, from the enzyme, you no longer demethylate. Not only this, but these genes are no longer upregulated as you try to induce your EMT, so it's inefficient EMT. Now, this is a dish experiment. What is happening in real human cancer settings? So we have been doing this experiment many times over, thanks to Christophe Ginessier and Emmanuel Charaf in Marseille, who identified tumors from humans that were characterized by this EMT profile, meaning metastasis, acquisition of drug tolerance, poor outcome. This is a residual tumor. We sort of CD44 high low cells. In the CD44 high cells, you see fibronectin that's up, you see transferrin receptor that is down, and yet ferritin is high. So question, how can you explain that ferritin is high, meaning more iron, if transferrin receptor is low? Because there is another pathway, and this is CD44 that triggers this. So the data show that in this process in vivo, in human, what dominates iron uptake is CD44. It is not transferrin receptor. Now PHF8 is up, the mark is down. That mark controls the expression of these two. And by the way, CD44 is one of the top genes, meaning that CD44 upregulates itself, unlike transferrin receptor, at the transcriptional/epigenetic level. Right. Now we looked at other tissue samples. So we looked at pancreatic ductal adenocarcinoma, also characterized by CD44 high, hyaluronate high in the stroma, highly metastatic and hard to treat. We sorted the cells, CD44 high low, more iron in CD44 high population, more iron in the PDAC compared to healthy tissues. And we see that there is more chemically reactive iron in the lysosomal compartment compared to the healthy tissues. You might ask, why is this so important to find out? Bob Weinberg did a screen, phenotypic screen, and most drugs have been identified out of screens. But we are chemists, and we are in the capacity to design new compounds, new chemotypes, new drugs that are unprecedented. And that is, to me, the beauty of chemistry. And so we came up with this idea. Is it possible to synthesize a lipid degrader, meaning a bifunctional compound, whereby you can control localization in lysosomes, and you can control the chemical reactivity of the metal that is abundant in the lysosome of drug-tolerant persistent cells. So this bifunctional compound contains two moieties, one that targets the plasma membrane, one that can target iron. This green part naturally does not go to the lysosome, let alone in proximity to the membrane, but when it is tethered to the red part, it does. So on endocytosis, the green part, which was outside the cell, ends up in the lumen. In the lumen, you have iron, hydrogen peroxide, molecular oxygen, and most importantly, pH 4.5 to 5, which is a typical pH you would want to promote Fenton chemistry. So you will form this iron reactive complex, but in proximity to the membrane. The proximity is very important. If the catalyst is remote, you will oxidize something else, but you want to target the membrane. You will form this iron 4 oxo complex that is highly reactive. This will hijack a hydrogen from PFAS at the membrane to form a carbon-centered radical that can react with molecular oxygen to form this lipid hydroperoxide. And this is where my good friend Marcus is chipping in with FSP1 and GPX4, supposed to take it out. And so the idea is to overload this enzyme with a compound that exacerbates the redox activity of the metal. Right, so we designed this compound. Literally, I swear this is true. It took me half an hour to draw on paper, and it took Tatiana in my lab 5 years to make. And it took 24 hours to challenge whether or not it was working. And I commend her because most people in my lab and in other labs would have just said, listen, I'm not going to do this. This is too risky. I'm not going to waste 5 years because you've got this crazy idea. But they did it. So now you treat your cells. So this moiety is fluorescent, so you can track it, colocalize with lysotracker, a lysosomal marker. This, which naturally doesn't go to the lysosome, now that it is tethered, goes. At 37 degrees, where endocytosis is working well, you see the staining. If you cool this down at 4 degrees, you no longer see the staining. This validates that it is taken up by endocytosis, unlike the Weinberg compound, which is ionophore and diffuses through membrane. So these are different compounds. One is lysosomotropic, and the other one is taken up by endocytosis. It's not the same. So we go back to the PDAC cells. We treat our cells, so untreated by lipidomics. You don't see much lipid peroxidation. If you treat with this compound phentomycin, which we named after Henry Fenton, the British chemist who discovered this chemistry in 1894, you will see lipid peroxidation. But if you co-treat with this lipophilic radical trapping agent evolved by life, especially to prevent this, you block this lipid peroxidation. So this is interesting. It validates our hypothesis of lipid degrader, but most importantly, the cell population that is depleted first is CD44 high compared to the others. Why? Because there is more iron in them. All fine. Now we went further down the path. We collected metastasis, which is a liver metastasis of pancreatic ductal adenocarcinoma, such that we could compare the two tissues. So this is just after surgery. Surgery At the hospital, at Curie, we get a call, Raph, do you want this metastasis? Of course. Can you collect some LC tissue as well as a comparison point? Same patient. So you can treat. So we dissociate the tumor and we treat dissociated tumor just post-surgery, but ex vivo. And what we see is that we deplete the CD44 high population from the tumor tissues, but we have no effect, no cell death, no lipid peroxidation in liver tissues. And yet hepatocytes are iron rich. The point is iron is not loaded in the lysosome. It's used someplace else, and that's why we don't kill them. Now, if you look at this population, you need to know that 15 years ago, Weinberg was isolating his cancer stem cells by treating the dish with Taxol. And if you use doxorubicin, you will see the same. Taxol, doxorubicin, irinotecan, 5-fluorouracil, cisplatin, you name them all. You have a dish, you stick this compound into the dish, and your cells are going to shift towards the CD44 high population. So this is a real problem. There is no drug today used in the clinic that is doing this, taking out CD44 high cells, the metastatic cells, the drug-tolerant cells, the cells that can adapt. Now, a few more data, and I don't know, Hugues, you tell me when I speak to... Okay. This is the iron part. What about copper? Of course, if you increase iron, you would expect that copper is also taken up. And this depends on what is available in the tumor microenvironment. When you induce your EMT, regardless of the tissue of origin, even mesenchymal tissues, sarcomas, they are mesenchymal, but they can become even more so mesenchymal, so it will be a mesenchymal to mesenchymal transition. You see increase of CD44 and you see increase of copper each time. These are 350 tumors that we profiled for the iron content. We are trying to establish a metallomic atlas in cancer to try to understand what's going on, how metal ions are being distributed between CAF, cancer cells, and immune cells. What we see across patients is that you will always find more copper and iron in the residual tissues after chemo and in the metastatic tissues compared to primary tumors each time. So this suggests that there is a causal role of copper as a driver of cell plasticity. Now I had to put this slide back in light of the lectures that were just before me. When you induce CMT, you see an increase of superoxide dismutase in most tissues, and you see a downregulation of catalase. So if you have both, then you will expect hydrogen peroxide to go up, and now you can study it. What is hydrogen peroxide doing in mitochondria? Not only do you have this, but you have the rise of copper, and we found that it was a mitochondrial rise. We know this because we extract mitochondria, and we find a chemical way to show that it was a copper 2 species and not a copper 1 species. Why do we care, you might ask? We do care because copper 1 and copper 2 do not have the same electron density, and you do not form the same geometry in complexes, meaning that you can design a compound that will selectively target copper 2 without touching copper 1. And why do you want to have this? Because you do not want to block the ETC. You just want to block the process catalyzed by copper 2, which is found only in the disease state, the mesenchymal, prometastatic, drug-tolerant cell state. We discovered that chemical reaction, hydrogen peroxide is chelated and activated by copper 2. This is basic Lewis acid chemistry. Hydride shift, you reduce hydrogen peroxide, you produce water, you oxidize NADH, you provide NAD+. So why would cells do this? Because de novo biosynthesis of NAD+ is not mitochondrial, it is outside, and it is more costly to import NAD+ and to biosynthesize it outside as opposed to using just basic redox chemistry. It is prebiotic, whereby copper is unchanged at the end of the reaction and hydrogen peroxide is very easy to produce. Right, so question, what can we do? Can we target and how do we inactivate copper 2 in mitochondria? Never done before. How do we start? We tried all compounds, used penicillamine, triantine, all the compounds used for Wilson disease. Nothing was working. People got depressed in the lab. And luckily, I did stumble on an old paper, 1929, Chemische Bericht. This journal does not exist anymore. Written in German, and the very first figure, there is just one figure. It is the chemical structure of 2-metformin, the type 2 diabetes drug, with copper 2. 2-metformin for 1 copper 2. And what is known about metformin is that you inhibit complex 1 in mitochondria, meaning somehow mitochondrial targeting, but the explicit mechanistic target of metformin is not known. What we also know is that patients taking metformin need to take 2.5 grams a day. Can you believe this? In a dish, you want to see a phenotype, you need 10 millimolar. No toxicity, but 10 millimolar is huge. But that 10 millimolar, to me, reflected this bimolecular complex with copper. If you want to saturate your copper and to form this complex, and if you want it to be stable, you need to use a large excess. This is what we call entropy. If you don't use 100 times the KD, you will not form this bivalent complex. And so entropy is fighting against us. But in chemistry, you can cheat entropy. The way to cheat entropy is not to have 2 molecules for 1, is to have 1 molecule for 1. So how can we do this? We just synthesize the dimer with 12 carbon, and 12 carbon is long enough to have this square planar complex with copper 2. If you use 4 carbon, it doesn't work because it is twisted. If you put 50 carbon, you put virtually some entropy back into your molecule. So if you want to have a compound for which the pharmacology is the same, you can add back your 4 carbon outside, and you end up with a perfect control. You've got the same molecular weight, the same polarity, the same everything. The only thing you affect is the capacity to bind to copper. So this compound is biologically active, and this one is not. We have the perfect control. Now you mix this with copper, you form this color, complex formation. You put this with copper, and you form this color. And this is how we know that it binds tightly. Now what do? We use 2 models of highly metastatic, drug-tolerant cancer models, pancreatic cancer, which is also known as the KPC, RAS and P53 out, and non-small cell lung carcinoma cells, which was nicely introduced by Marcus. RAS mutation for most of them, the most aggressive forms, and P53 out. You can induce your EMT, meaning the acquisition of stemness, by treating this cell with TGF-beta, and that stemness confers this drug tolerance. So you would see CD44 going up using TGF-beta. Not only that, but you see copper going up as well. That is not known, this is not published. Now, when you induce your EMT, you will see a loss of ikaderin, which is the protein that allows cells to attach to one another. So, of course, if you take this out, then the cells are more susceptible to escape and to migrate. And you see upregulation of this mesenchymal protein that are associated to cancer metastasis. But now, when you co-treat with this compound LCC12, also known as superformin or subformin, you block this profile. Meaning that if you take out copper-2 in mitochondria, as you try to induce your EMT, you take out alpha-ketoglutarate, you take out acetyl-CoA, you cannot reprogram the epigenetic landscape, and you block the transcriptional programs regulated by slug, twist, snail, ZEB1, all of which known to promote the EMT and that are required for metastatic dissemination and drug tolerance. You see this here, and you see this in lung as well. So we cherry-picked those because we know them, but we looked by mass spec again at those. So the KPC model from 2000, and this triple neg system, and you see the same. You induce your EMT, you see this upregulation, and you co-treat with LCC12, and you block this upregulation. So the idea now for us is to challenge this compound in vivo, to try to see whether we can recapitulate this phenotype in vivo, knowing that this drug block activation of macrophages in acute inflammation in vivo. So we know it is active in vivo. And so we have this anti-cancer property in vitro, we have the biological activity in vivo, and next step is to see whether or not we can, by blocking cell adaptation, we block metastatic dissemination and we increase survival. And these experiments are ongoing. So listen, CD44, which was coined as a cancer cell marker, as a poor prognosis cancer marker, as an immune cell activation marker, as a developmental marker, it's not a marker of anything. If it marks one thing, it is the capacity of a cell to adapt. When you see upregulation of CD44, it means that your cells are adopting a new phenotype. Plain and simple. How does it do so? By promoting iron and copper uptake, which will reprogram the metabolic epigenetic landscape and transcriptional program, which explain how these are different identities. Lysomal iron 2 and mitochondrial copper 2 are druggable. Now, it is very important to point this out because there are many chelators of metals used in the clinic and nobody understands how it works. If you compare deferoxamine and deferazirox, both of which are used in the clinics, side effects are different. And clinicians, they have no clue. Why are these different? Because they do not target the same spot in the cell. And so you might have the global effect of iron depletion that you are seeking for, but at the same time you induce side effects that are linked to the organelle that you target. So knowing that you can target a metal as a degree of oxidation specific and in an organelle that you know is highly desirable. Lysosomal iron is a trigger of ferroptosis. Cell state defines cell death modalities. This was shown by Marcus and many other people. Cancer, I'll pass on this one. And now this we have shown in the context of inflammation. If you control adaptation or activation of macrophages, you reduce acute inflammation in vivo and you prevent sepsis. And so back in the days, Linus Pauling with the sickle cell anemia mutation said, if you correct the genetic defect, you correct the disease. We didn't know what the gene was about. It was 4 years before double helical DNA. And David Liu, 70 years later, using CRISPR, has shown that actually if you correct the genetic mutation, you reverse sickle cell anemia, 70 years, which convey the idea that diseases are genetic. But we know that the metastatic aspect of cancer is non-genetically driven. And so the question that we have is, if you control cell adaptation, can you also confer therapeutic benefit independently of genetic alteration? So we have shown this in inflammation, and we will try to see whether we can see this in cancer. And with this, I would like to thank you all so very much.